Hello guys, today we are doing question 4, which is basic agricultural genetics. Yeah, let's roll on. 4.1, the crossing of black BB male farm animal and a white WW female farm animal gave rise to a heterozygous gray offspring in the F1 generation. The same offspring of the F1 generation were allowed to breed through inbreeding. Their offspring in the F2 generation had a phenotypic ratio of 1:2:1, and they give us an illustration showing the crossing. Okay, and before I can get into answering the question or reading the question, because there is some terms that we need to know first. So we have a new member in the house. Agricultural equivalents have a new member. Her name is Memo Olivia, and yeah, she's going to give you a lecture for the introduction lecture to agricultural genetics. Then yeah, let's give her an opportunity so that we can learn from her. Drum roll please.
Hello learners, I am Nan Olivia and welcome. This is Agricultural Sciences class and I'm going to give you an introduction into agricultural genetics, and feel free to ask questions and enjoy. Basic agricultural genetics. We will look at genetic concepts, monohybrid and dihybrid, inheritance, the pattern of inheritance, variation and mutation, selection and genetic modification engineering. Okay, let's see. Genetic concepts. Genetics is the study of heredity, showing how the characteristics of parents are transferred to the offspring. Offspring is a progeny. Gene is a unit or a segment of DNA that codes for a specific characteristic, for example, shape of a fruit. Alleles are different forms of a gene. Locus, a position where the gene is located on a chromosome. Heredity is how the characteristics are passed from generation to generation. Monohybrid inheritance, a genetic cross involving only one characteristic, e.g., flower color. Dihybrid inheritance, a genetic cross involving two characteristics, e.g., flower shape and flower color. Homozygous to homozygote, an individual with two identical alleles for a characteristic, and both allele are shown in the phenotype. Heterozygous, heterozygote, an individual with two different alleles for a characteristic, and only the allele that is dominant is shown in the phenotype. Meiosis, a cell division that results in 4 daughter cells that are haploid. In animals, it is called gametes. In plants, it is called spores.
Now, because we know what these terms mean, so we'll go back to the scenario. The crossing of black homozygous dominant male. Okay, homozygous dominant male. So this BB means homozygous dominant because that's 2 capital letter B's. Okay? Then also the white female is also homozygous dominant because it has 2 capital letter W's. Okay, so a alternative name for this homozygous is purebred. Purebred is the same, means the same thing as homozygous. Okay, then the animal gave rise to a heterozygous gray. That means replacing a heterozygous offspring. Okay, then if that offspring is heterozygous and both parents are homozygous dominant, then what is the type of dominance here? Because the offspring is heterozygous gray, but we know that the parents are what? Are black and white. And this offspring didn't take any of the parents' color. This offspring has a mixed blend of uh, the color of both parents. Okay, so this shows us that okay, this offspring has a new color that is um, not similar to the color of the parent, because the parents have black color and white color, but this offspring has gray color. That means this type of dominance is incomplete dominance. Okay, so why are we saying it is incomplete dominance? Okay, this is because it gave us a mixed blend of color, or an intermediate of black and white, which is gray. Okay, so in that situation, when there is a mixed blend, that's when we have incomplete dominance. Okay, then which is 4.1.1. Okay, 4.1.2, give 2 reasons to motivate your answer to question 4.1.1. 2 dominant alleles get to express themselves in the genotype. 2 dominant alleles get to express themselves in the genotype, and as a result, we get a mixed blend. Okay, as a result, we get a mixed blend. We get a mixed blend to form a new color when expressed in the phenotype. 2 dominant alleles get to express themselves in the genotype, and they result in a mixed blend to form a new color when expressed in the genotype. Then 4.1.3, complete a diagram and write down the missing genotype at A, B, C, E, and G. Okay, so for us to answer this question, we need to draft this question somewhere. Okay, so I will post this thing. Okay, yes, we post it. Yeah, so it should be like this. Then yeah, this is how we should do it. So we should draft this question somewhere so that you know what is going on. So the phenotype, we have black and white, and we were told that this black is homozygous dominant because of 2 capital letter, and it is caused with a homozygous dominant female, which is white color. Okay, then the genotype is this alleles, dominant BB, homozygous dominant B, and it is caused with a homozygous dominant W. Okay, then this genotype is going to go through meiosis. Then we are going to get our gametes. This is the gametes. Dominant B is separated during meiosis. Then we get dominant B and dominant B, and we crossing it with dominant W and dominant W. Okay, these are our gametes that are separated during meiosis. Then okay, then you can see what. Okay, C is equals to W. Okay, C is equals to W, and D is equals to W. B is equals to C is equals to W, D is equals to W, E is equals to B, F is equals to B. Then G, that's okay. Let me work out G. G, then after we have commit formation during meiosis, then it goes through the process of fertilization. Then first cell generation, you have the genotype G, which is BW, 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 BW. They are all BW. That means they are all heterozygous offspring. BW, BW. Okay. Then this BW is going to be the first generation, né? So that means we got our answer, which is G. That's BW, heterozygous BW. Then that's all for question 4.3. 4.2. Let's see. Okay, 4.2. Let's see. A recent development in the improvement of maize is a genetic modification that makes it resistant to the maize stalk borer. A soil bacterium, Bacillus thuringiensis, which is Bt, naturally produces a toxin which is a poison that kills the maize stalk borer. Genetic engineering techniques are used to transfer the Bt toxin gene from the bacterium to the DNA of maize plants. 4.2.1 Identify 2 potential benefits of this genetically modified crop. It prevents the maize stalk borer from eating the maize plant. Né? Then another point is that it helps to reduce damage to the maize. It improves the yield and the quality. Okay, it improves the yield and the quality. Meaning it stops yield losses. Agree? If this thing is, if this maize stalk borer keep on eating the maize, that means there will be yield losses because they won't be able to produce the required yield that they aim to produce, because every day they go, okay, want to produce 500 tons, so that means that they won't be able to produce 500 tons because that means stalk borer will be contributing to the yield losses. That means okay, if we have this GM that is resistant to this maize stalk borer, then okay, that means it helps to reduce the damage to the maize, and at the same time it improves the yield and the quality of our crops. Yeah? It stops the yield losses. 4.2.2 Explain how the bacterium in the scenario above is used to modify maize plants genetically. Okay, so explain how. Alright, the key there, explain how. So they want a detailed step-by-step of how this is done. Okay, so they need to so here you need to have knowledge of how agrobacterium tumefacius is used in plants to insert other DNA into crop plants. Okay, we need to have knowledge of how agrobacterium tumefacius is used in plants to insert other DNAs into crop plants. So agrobacterium is one of the techniques used in genetic modification in plants, yeah, as we know it. And yeah, but here they gave us another bacteria called Bacillus thuringiensis. Bacillus thuringiensis. So you just need to explain um, how that DNA is extracted from Bacillus thuringiensis bacterium and how it is transferred, and then how it then transferred to the maize plant. Okay, so yeah, then you need to give them the result of that step. After that, you need to give them the result of that step. You repeat that. You just need to explain to them that the DNA is extracted, okay, or it is removed from the Bacillus thuringiensis bacterium and is transferred to the yeast plant. Okay, after telling them that how to do that, then you give them the result of that step. Okay, of what that will mean for the plant. If we extracted the DNA from the bacterium and transferred the DNA to the yeast plant, then what does that step due to the maize plant? What is the result of that technique, or what is the result of doing all of those things? Okay, so as a result, the maize plant develops resistance to the maize stalk borer. Okay, the maize plant develops what? Resistance to the maize stalk borer. They wanted to know that. Okay, then for this question, it is 3 marks. Next, we're going to get 1 mark for the how part. How do we transfer this DNA, or how do we get this DNA so that this DNA can end up in the maize plant? How? Okay, the how part. Then another mark is for mentioning the bacteria. You need to mention the DNA is extracted from this specific bacteria, Bacillus thuringiensis bacteria. Just by mentioning Bacillus thuringiensis bacterium, you get 1 mark. Okay. Then the last mark, you're going to get this last mark, which is 1 mark for the result. What is the result of that? Okay, that's where you're going to tell them that if the result, the maize plant develops resistant to maize stalk borer, then you get 1 mark. Then the total is 3 marks. 4.2.3 explain 2 negative effects of GM crops on the environment. This question is what needs you to give 2 negative effects. Right? So, number 1 is that it creates superweeds. The creation of superweeds, it creates superweeds, and B, it kills useful insects or beneficial insects. Okay, it kills useful insects or beneficial insects. 4.3. The flowchart below is a schematic representation of line breeding. Okay, so as we can see, we have these lines. Okay, we have lines going from 13 then to all these numbers until x, right? Then let's go to the question so that we can see what we're looking at here. 4.3.1 identify 2 common ancestors of individual S and D in the schematic representation. Okay, so 2 common ancestors of individual S and D. Yes, S and D. Okay, two common ancestors. Query, they want the common ancestor. Query, the ancestor that gave rise to F and D. Okay, so common grandparents that give birth to their parents. So they just want those grandparents that give birth to their parents. So it's like a family tree. Ne? But this one is horizontal. It is horizontal instead of being vertical. You know, the family tree is vertical, where we start from the top, where we have the grandparents, and the parents, then the offspring. So, but this one is vertical. It is horizontal, not vertical. So, yeah, can be a little bit tricky because we know the vertical one, but this one is horizontal. So 13 here is the grandparent that gave rise to 6, 2, and the letter S was born. Okay, then the letter S was born. Then another grandparent is 5. Yes, 5 is a grandparent that gave rise to 4. 4 here. See the arrow that that goes to 4, from 5 to 4. Okay, if the grandparent that gave rise to 4, then the letter D was born. Okay, okay, right. Then the grandparent 7. Okay, it's the grandparent 7 here. If the grandparent 7 is a grandparent that gave rise to 3. 3 is 3. Then 3 give birth to letter D. Then that's where letter D was born. Okay, that means we have 13, 5, and 7. Yeah, that is two common ancestors. Okay, you just need to mention the numbers. You don't need to name them. Just take the numbers. These numbers represent the ancestor. Okay. 4.3.2 explain 2 ways in which livestock farmers could benefit from upgrading by using a pure breed in their commercial crossbreed herd. Okay. So, okay. A is that pure breed will improve the characteristics of the livestock with rapid results of up to 50% improvement in the first generation. Pure breed will improve the characteristics of the livestock with rapid results of up to 50% improvement in the first generation. B is that it can bring dominant useful traits and mask the defective traits to improve the livestock. It can bring dominant useful traits and mask the defective traits to improve the livestock. So a farmer planted maize seeds with the same gene for height and yield on a field A and field B. The seeds in field A produce tall maize with 2 to 3 cobs, while the seed in field B produced short maize with 1 cob. 4.4.1 Identify the genetic phenomenon referred to in the scenario above. So the one-day genetic phenomenon. Okay. So here we can see the difference between organisms in a population. For this to happen, it was caused by the external causes. The external causes made this to happen, which is the environment they were raised in. Okay, that was the external causes, the environment that they were raised in. So here we have field A and field B. So these two fields affected the yields of the two maize plants. Okay, that's why we ended having the maize plant that was grown in this field, which is field A. This maize plant was tall, and the maize plant that was grown in field B was short. Okay, we ended up having short and tall plant. So this is variation. So the genetic phenomenon is variation. Guys, this cannot be confused with epistasis. Okay, epistasis is not what they want here. Here they want variation. They're talking about variation. So the genetic phenomenon that is happening here is variation. 4.2 name two possible external causes of the phenomenon in question 4.4.1. So here they want the external causes. What can cause this to happen? Okay. So this can be caused by the environment. Other field A and field B have different environment. And yeah, the soil, the environment. So the soil in that environment. And B, the climate. The climate in two different fields when they're the same. So the climate affected that. I get the temperature, so all those things, environmental factors. I mean the climatic factors. The climatic factors, temperature, all those things. Okay. Temperature affected the growth, because the other one was tall and the other one was short. 4.4.3 select one factor of importance of this genetic phenomenon in plant breeding. So here they want the importance of variation. Okay, so we will tell them, okay, variation helps to select parents with good characteristics. With variation, you can select parents with good characteristics. And B can also say it helps to ensure crop variation. Yeah, crop variation or crop varieties. Okay. Yeah, 4.5 next. Hereford cattle which produce heavy and voluminous are mated with woolly cattle which are hardy and disease resistant. 4.5.1 identify the animal breeding system used by the farmer above. Then okay, this is crossbreeding. Okay, this is the crossing of two different breeds. Hereford cattle and Nguni cattle. Okay, two different breeds of the same species. Either Hereford cattle and Nguni cattle are the same species. Okay, so yeah. Then 4.5.2 give a reason for ants. Okay, is the crossing of two different groups of the same species. For example, here we have two different breeds, Nguni cattle and Hereford cattle, which have two different breeds. Yeah. Then for this question, it was needed. So they wanted a reason that is related to crossbreeding, like related to the definition of crossbreeding. Okay, yeah. So if you say two different breeds with two different traits are crossed, no, that's wrong. You can't say that. You can't say two different breeds with two different traits. That is not correct. What you have to say is the crossing of two different breeds of the same species. Then you get number. Open pattern 2. Let's open pattern 3. Yeah, 3. Give 2 reasons why the breeding system above is advantageous to the farm. So it is advantageous to the farmer because it produces hybrid vigor. Okay, hybrid vigor basically is when offspring have better performance than the parent. So that's the result of crossbreeding. Okay, so we end up having these offspring that have better performance than the parents, so they can perform better than the parents. Okay, that is hybrid vigor. We can have increased genetic variation in the head. Okay, so this increases genetic variation because this crossbreeding will make our head to have different kinds of breeds. Okay, yeah, so this increases genetic variation of the head. So you can't have uniformity in the head, alright? Yeah. So 4.5.4. The two disadvantages of mating the progeny of Hereford cows with the same Hereford parent bull. So if we cross this offsprings of the Hereford cows with the Hereford parent bull, which is the parent, which is the father of that offspring, that will lead us to what? Inbreeding. Okay, then now here they tricked us. They want the disadvantages of inbreeding. Okay, so the two disadvantages of inbreeding, it will lead to inbreeding depression. Inbreeding to depression. Okay, that's point number 1. It will lead to inbreeding depression. It leads to inbreeding depression. And again, it can result in lethal genes present. Okay, can result in lethal genes present. Can result in lethal genes present in the offspring. You know that the offspring can end up having lethal genes. Okay, lethal genes, and these lethal genes, they are harmful genes. Alright, so yeah, and also a bonus answer, we can say the offspring can be defective. Okay, the offspring can have deformities. You know, the offspring can be defective, and yeah, defective. I don't know, defective basically, that's when we are talking about defective traits in the offspring. Okay, as a result of inbreeding, where the offspring have 5 legs or one. So those defective traits that make it have 5 legs or one. So those are the defective traits. So yeah, deformed offspring. That offspring can have difficulties, maybe difficulties working, difficulties raising, all those things. That's why we say the offspring can have defective traits, or we can just say deformed offspring. Deformed offspring is correct. That's the correct term for this thing that I'm talking about. The correct term for the formed offspring. So that means when we can say can lead to formed offspring, or we can say can lead to inbreeding depression. Okay, and also you can say it can result in lethal genes being present in the offspring. 4.6, the accuracy of an estimated breeding yield, which is EBV, is an indication of its reliability and the risk entailed when a specific trait is selected. Traits with a low accuracy are highly unreliable. The table below shows the characteristics with heritability and accuracy percentages. Let's pause there. Okay. We want to clarify something. Tricked us. What is a trait? A trait is a characteristic. So they go hand in hand as similar terms. The traits are characteristics. Characteristics are traits. It's the same thing. Characteristics: death weight, fleece weight, slaughter weight. Reachability percentages, accuracy percentages. So when we look at our table here, we see okay, fleece weight is the one that has the most reachability, which is 68. Okay, and but it has a low accuracy of 40%, which raises some red flags about it. Okay, and the other ones you can see, okay, they're all about they're all having high percentages, like the death rate having heritability of 65 and 80, accuracy, then slaughter rate 61, reachability and accuracy of 50% high. Okay, fine. This is interesting. Already it is interesting. Let's get into question 4.6.1. Identify two characteristics the farmer is likely to select in the table above. So two characteristics. We want those ones that have high percentages. So we have 65 and 80. Yes, I can take this one. That weight, Lagara box. Okay, we take that weight. Fleece weight, no, we can't take fleece weight because of this red flag of 40%, which is low. No, don't take fleece weight. Slaughter weight, yes, you can take slaughter weight because it has 61 and 50. So birth weight and slaughter weight. 4.6.2. Why? Two reasons for your answer. So the reason for this is because of what? It's because the birth weight has heritability of 65% and accuracy of 80%, which is high. Okay, so this shows that they are reliable. Okay, they are reliable. And also slaughter weight. Slaughter weight has reachability of 61 and accuracy percentage of 60%, which is high. So this shows that it is also reliable. Okay, we can't take fleece weight here because fleece weight has high heritability of 68 but accuracy of 80, which is not good because both of them used to be high. But with birth weight has reachability of 65% and accuracy of 80%, which is good. And slaughter weight also has 61 and 60, which is also good. So that's why we took this one. Then 4.6.3. Give two reasons why the estimated breeding value is important in a breeding program. In a breeding program, why is it important? It is important because it shows us the prediction of the performance of the progeny for a particular characteristic. Okay, let me try to put this in a more simple way. Progeny means an offspring, né? So let me try and break it down. So it helps us to predict the performance of the offspring for a particular characteristic. Okay, so prediction is guessing on what will be the characteristic, what characteristics will be in the offspring. That's the prediction, guessing. But now we are guessing using data. I want to data this data that we get from heritability, those percentages. The higher it gets, the more reliable it gets. Okay, the more reliable the result. Okay, also the accuracy part of it. Right. So the prediction of the performance of the protein for a particular characteristic. So it shows how more or less the offspring will be for a specific characteristic. Okay. Yes. And B, we can also say that it indicates the characteristic to select for breeding purposes based on the performance. It shows us and indicates to us the characteristic that we can select for breeding purposes based on the performance. Okay, so eventually this helps the farmer to select a characteristic that is reliable and will produce the expected result. Okay, will produce the expected result. And that's it for this paper.